Hello students, welcome to Learner's Planet. Let us continue with the chapter Reproduction in Animals. Well, in the earlier session, we were discussing about the sexual reproduction, right? Sexual reproduction, that is the mode of reproduction in which the sexual parts of the body are involved. It begins with the fertilization of the gametes, right? And the gametes are produced by male and the female organisms. Now, if this reproduction has to take place, there should be the fertilization, right? That means the egg cells and the sperm cells, they will undergo the fusion process. Now, students, this fertilization, it can take place either inside the body or outside the body. But when we are discussing about human beings, definitely it is going to take place inside the body. They show the internal fertilization. But there are some people which are there are the people who are facing the problem with the process of fertilization. The sperm is available, but as well as the ovum is available, but still fertilization cannot take place. Reasons are unlimited. There may be some hormonal changes, the body is not properly functioning so as to proceed the process, and there actually we cannot I cannot explain it right now as it is not in your syllabus, but there are many different reasons because of which the fertilization is not taking place. Now, do you think that these people will not have the babies forever? It was so in the earlier period. But nowadays, there is the opportunity, there is the application by which the process of reproduction can take place, or the fertilization process can take place artificially, and thus the reproduction can be continued. And that technique is known as IVF, that is in vitro fertilization. In today's session, we are going to discuss about it briefly. So, in such a condition, see, the women, they are unable to bear babies, this is for sure. And one of the reasons given here is that the sperms cannot reach the egg. And if they are not able to reach the eggs, definitely fertilization cannot take place. Or we can say the sperm count is very less, right? Due to which it is not able to reach it. Or there are some hormonal problems. Or the mature egg is not released by the female body. Isn't it? There are many different reasons. So, to overcome this problem, in vitro fertilization is actually this technique is followed nowadays so as to produce the babies. And such type of babies which are produced as a result of in vitro fertilization, they are known as test tube babies. See, what is the meaning of this in vitro? In biology or in science, two terms are used, that is in vitro and in vivo. In vivo means what? Inside the body or inside the cell. In vitro means what? Outside. So in this technique, what is actually happening? Fertilization is taking place outside the body. Normally it occurs inside, right? But in this technique, the fertilization is induced outside the body. And now for this, what should happen? The sperms and eggs, they should be present outside the body, isn't it? So in this technique, this is the way that how it is actually followed. From the male and the female body, the gametes are extracted out and they are allowed to undergo the process of fertilization. How it happens? Let me explain you. Now for this purpose, of course there are some formalities that should be followed initially. Now further, what happens? The doctors, they will collect the freshly released egg and the sperms. Now where these egg and sperms are present? Eggs are secreted or they are produced by the female reproductive system. So from the uterus, right, these eggs are extracted. or from the female reproductive system, that is fallopian tube or this uterus, these mature eggs which are released from the ovary, they are collected out. So, and I'm sorry, on the other hand, sperms are collected from the male body. So the semen which is ejaculated out from the male's body, right, that semen is collected. And now these eggs and the sperms, now they are allowed to fertilize. They are kept together in a medium and they are incubated at a particular temperature. Right. So favorable conditions are maintained, and if these cells they comes in contact with each other, they are going to get fuse. Right. And therefore, in such case, in case if the fertilization occurs, if the fusion is going on in the right way, then the zygote is produced. And of course, this zygote, if it is produced, it will be maintained further in the in vitro conditions itself, at a particular temperature and the favorable medium and with all favorable conditions, right? And this process will take place around a week. This is the incubator where these fused eggs are supposed to be placed. Now, of course, after that week of incubation, the cell division will continue and the growing embryo is now placed in the mother's body, that is in the uterus. And further development will take place in the uterus of the mother body in the natural way. And later on, the baby is born like any other baby. Right. So this is how you can see that how these gametes are collected. They are placed in the Petri plate and then they are incubated here, isn't it? And of course, then the embryo is placed back in the mother's body. This is how the in vitro fertilization is working. Now, the chances of success are, of course, it is not predicted correctly. It may be successful or it may be a failure. So, all depends upon the conditions in which these eggs or the sperms are present and other physical and other chemical conditions also. But definitely, if the zygote, if the process is going on in the right way, the test tube babies are a good option for those particular couples those who are facing the problems with respect to such type of things. That is with the fertilization process, and thus it is a solution for them to get rid of their problem. So students, now you can see that how this application is helping the couples actually. Besides this, these type of techniques are also used for different purposes, like for improving the quality like let us say, for example, in the some of the breeds or the dairy organisms, right? Dairy animals, like those who are producing the milk, right? Those milk producing animals, let us say, for example, I will just tell you, if it is cow, right, the Indian breed is resistant to diseases, right? And the foreign breeds, they are having good lactation period, but they are not very much resistant to the diseases. So these two organisms, if they are allowed to mate with each other, the new organism which will be produced, they may have the properties of both these organisms, right? So this type of combination in the new organisms will help in improving the quality of the quantity of the milk produced, as well as the resistance which was developed in the organism, so that for a long period of time fresh healthy milk is obtained from these animals, right? Besides this, there is a technique which is known as cloning, where such type of things are also applied. And you know what is cloning? Let me explain this thing. Well, students, this cloning, it is the production of an exact copy of a cell. We can say copy of a cell or a complete copy of an organism. How? A copy is produced in an artificial manner, right? And it is done by using some living part of the body. So this is how the cloning is actually done. Cloning is basically copying. right? And when we are using this term with respect to this subject, then of course it means the production of an exact copy of the living organism. And this living organism, it can be a single cell, that is, it can be a unicellular, or it can be a multicellular organism. Now, of course, if a unicellular organism is considered, then definitely each organism, these unicellular organisms, they are reproducing by the asexual method, isn't it? So over there, the system is naturally supporting the cloning over there. Whatever genes are present in the mother cell, they are transmitted as such in the next generation, right? But when we compare it with the higher organisms, then over there are some complications. The organisms which are produced, they are not the exact copy of the living of their parental organisms or the parent generations. You can observe yourself. We are produced from our parents, right? But are we really the copy? Are we really the exact carbon copy of our parents? No, we are not. So, if the exact copy of such organism is to be produced, then it is done in this artificial manner by using some living cells of these organisms, right? So, the very first clone which was produced successfully, it was Dolly. You must be knowing it. It is the sheep, right? So this clone, the first clone produced was Dolly. And who was that great scientist who has done this successful experiment? It was Sir Ian Wilmot and his colleagues, of course, at the Roslin Institute in Edinburgh, Scotland. Uh, So now I will actually explain it to you in a very brief manner that how this Dolly was produced, right? So during this process of cloning of Dolly, a cell was collected from the mammary gland of a female Finn Dorset sheep, right? And you can see this diagram here. Simultaneously, an egg was also obtained from a Scottish black-faced eve. So you can see this picture here. This is the white-faced sheep, right? That is the female Finn Dorset sheep, right? And this organism, or the from this sheep, the mammary cell was extracted. Fine. Further, on the other hand, the egg of this particular sheep, that is black-faced, the egg was released from this organism. Fine. Now, what was the purpose of this thing? During this process, what these scientists wanted to be done is they wanted the features of this white-faced sheep organism to be transmitted into the offspring of these black-faced sheep organisms. And for that, what they have to do? They have to transmit the genes which are present in this white-faced sheep into this another one, right? So how is it possible? It was possible as they removed the mammary cell of this organism, and then they removed the nucleus of this cell, and transmitted transferred it into the egg of the another female, right? See, you can see here with this thing, with the text, of course, initially the nucleus was removed from the egg, right? Where is the egg in this picture? See, this is the donor organism which has given us the egg here. From this, by a particular method, the egg was released out. Now the cell is enucleated, right? Now further, the nucleus of the mammary gland was now inserted into the egg of Scottish black-faced Eve, whose nucleus was removed, right? So here you can see that the two cells, they have to be fused together, and this was done with the help of the electricity. Now of course, the high power electricity is not used for such purpose. Fine. It is accordingly used in the experiment, and during this, what happened? The two cells, which cells? The enucleated egg, the egg from which the nucleus has been removed earlier, and this particular cell which is obtained from the mammary glands of the sheep, white-faced sheep, and then they were allowed to fuse together. As a result, a new egg cell was produced in which the nucleus of white-faced sheep was present. Further, now such fertilized egg was allowed to grow further, and later the cell division was continued. It was maintained artificially for a particular period of time, and then this growing embryo was implanted into the black-faced sheep. Right, and further the normal cycle was going on, and at the end of this gestation period, the baby sheep was born. It was born, sorry, and this baby sheep or this lamb was having the white face. So now you can see that this white faced sheep was actually the clone of the this particular white faced sheep. So this is how the process was actually performed. Of course, it is not as easy as we are discussing. It is a complicated process, and it takes a lot of time to conduct such type of experiments. But definitely, after such a great work, after such a great hard work, the final product was there in the form of this clone, that is Dolly. Dolly was of course found to be absolutely identical to the Finn Dorset sheep, from which the nucleus was taken. So now you can understand that it is this nucleus which carries the information from one generation to the another, because no further other changes were done during the process, isn't it? Well, this is how the development was continued and the Dolly was produced, and unfortunately in 2003, that is February 14th 2003, the Dolly, she died. It was because of certain lung infection, and this was the end of her life. Well, students, beside these type of applications, some very naturally existing interesting facts are also observed. There is a division of labor in some organisms. Now, I know that division of labor is not something difficult for you to understand, but I'm not talking about the division of labor within the individual body or the individual cell. I'm saying division of the division of labor is observed in a group of organisms or in the population of an organism. The sexually reproducing organisms, they show some interesting division of labor among their group, and that is of course related to sexual reproduction also. I would just like to give you one example for this thing that is related to the honeybees. Let me explain that how division of labor is observed in this particular organism or the colony of this organism. So students, in the honeybees, as I said, division of labor is observed. There is an interesting organization. In a hive, thousands of honeybees are living. There is an entire colony within the same the hive, right? And in this colony, the organisms, they are having their particular role. Let me just explain you with the help of this example, honeybee and the bumblebee. See, there is a single female organism which is known as the queen, and this queen is required for laying eggs. This single organism in this entire colony is actually laying thousands of eggs, which will further mature into the organism. They will grow into the matured organisms. Right. Now, of course, in similar species also such type of pattern is observed with some mild changes. But of course, there is a single queen which is responsible for laying the eggs. Right. Then further, what about the other females? All other females, they are known as the worker bees. And these worker bees, they are responsible, or they are taking the job of taking care of the larvae or these eggs which are hatched or which are produced by the female or the queen bee. Besides that, it also helps in building up of the hive, its maintenance, maintaining the temperature inside, feeding the queen bee so that she can lay so many eggs. So in a particular colony, there are almost 10,000 to 50,000 honeybees, worker bees in a hive. And if we compare it to the bee colony, then in that case there are around 50 to 400 organisms or the worker bees, depending on the type of species. Fine. Then further, there are the males also. These males, they are of course required for mating purpose or for providing the sperms. Now in this case, what happens, uh, the eggs which are laid by the queen, if they get fertilized, then they will become the female, and if they remains unfertilized, so they will become the males. And these male organisms in this hive or in the honeybees colony is known as the drones. So you can understand that how the work related to this reproduction process is also divided into the different organisms in some of the species. Isn't it so interesting? I think it is so. And I hope you have enjoyed all these things very well. Further, we are going to continue in the next session. And then over there, we are going to discuss about the asexual reproduction. Till then, thank you so much. Have a nice day. Goodbye. See you soon.